Okay, hello everyone. So welcome again to the special session on omics data compression. So we will have now 3 more talks until 6 p.m. And I'm glad to introduce our next speaker, Christian Iseli from the Swiss Bioinformatics Institute.
Thank you, Idoia. Thank you to the organizer for inviting me for this talk. I'm Christian Iseli. I work for the Swiss Institute of Bioinformatics, which is an organization that was founded 20 years ago this year, mainly to first federate bioinformaticians in Switzerland and also to provide a home for databases like the Swiss protein databases and a means to get funding to these kind of efforts. And I work in a group called Vital IT, which was founded by Victor Jongeneel, which some of you might know 15 years ago in 2003. And it is a group that started basically as a high-performance computing center for life sciences, and nowadays it's more called a competency center, so it grew to about 40 people, where we mainly collaborate. It was founded mainly by the University of Lausanne, the University of Geneva, and the EPFL, which are the 3 main universities in the western part of Switzerland, which is the French-speaking part, and we mainly collaborate with these groups to provide bioinformatics supports, either through machines and storage, or more mainly now through providing bioinformatics support, participating in projects, and helping basically people when they don't have in their own lab the bioinformatics support they would need. The idea of the talk today is to provide some insight into what were our main thoughts or so when we were asked to participate in this MPEG consortium to provide some sort of standardization to store and compress data formats. So I will try to give a little story or a history of what were the various things that came into our minds when we tried to provide input for this effort. And basically, I start the story with this H-Brain SysBio project, in which we started participating back in 2012, which was about trying to correlate images they got from people who were diagnosed with Alzheimer's disease, compared with some people who had normal, who were not affected with this, and another cohort or people who were sort of on the way to the Alzheimer's. There were these 3 cohorts. It was about 800 persons in this study, and they had various brain imaging, but also at the time, and that was new to us, they had whole genome for all of these 800 patients. And the question was basically and this was done with a group called ADNI, which was based in the United States. And the question basically for us was, how do we actually get access to this data to process them locally? 800 full genomes, at the time they were stored as BAM files. Each BAM file was about 100 gigabytes, so the BAM file is a way to store genomic data. You've seen this before. And for each of the patients, basically they had the BAM file, they already had done the calling, so there was a VCF. And it weighed 85 terabytes, which somehow we wanted to get across from San Diego, where it was originating, to Switzerland. And that was actually quite a problem. And at the time, the only practical way we found was to ship a whole machine there in a big box, have the people there actually put the data in the machine on the hard drives in an organized way. And once this was done, to ship the machine back to us in Switzerland with, of course, all the funny things at the customs, explaining what was in the machine, etc. And that happened in 2014, in the fall of 2014, when we actually got the data. And we thought, that was one of our thoughts at the time, this was really cumbersome, and if these initiatives have to come true, that we will nowadays sequence the whole genome of all patients in the hospitals and analyze and store these things and keep them around for the whole lifetime of a patient, well, that was an interesting problem. And the various things we wanted to do with, well, first we noticed when we wanted actually to reprocess the data to do some further analysis, it wasn't actually that easy to re-extract the original FASTQ from the BAM, because it's not clear when you have paired reads which one goes with which one. You have to do some analysis. Before being able to use the file, we had to index them, and indexing each file took about 20 minutes. So since we had 800 of them, that already was another issue. And when you re-extract the FASTQ, it's actually 300 gigabytes, which you can back down to the 100 gigabyte if you compress with gzip, for example. But again, that takes time. And so, and also another issue that we didn't quite realize at the time, but if you apply the standard variant detection tool, if you reapply it, for example, on the re-extracted FASTQ, you didn't get the same output, the exact same VCF as they had provided, even if you use the same tool, because they are apparently not completely deterministic. So that also was a bit of a surprise to us at the time. And we decided we wanted to redo some of the analysis, but using our own tool. So we have some homegrown tools or others we wanted to try. And simply processing this took, it could take several days per sample sometimes. So we thought, yeah, we need some better tools for that. And we wanted to make sure, for example, if we wanted to always have a consistent answer, it's always the same output for the same file, we thought maybe some exhaustive tool would be useful. So that's why also we try to do our own. And basically, we didn't always, depending how you filter, how you set the quality score, if you decided to make more stringent filter or not, you don't always get the same answer, basically. And we think this problem is not going to go away, and depending on what you're looking after, if you want to be really exhaustive and you want to minimize false negatives, make sure you get all the possible answer, maybe you filter some way. But if you only want the things of you're completely sure, you don't want to have any false positive, then you filter differently, maybe you need to reanalyze differently. So really a way to store and get at the data is important in that case when you want to reprocess. And it's more like you need to define some kind of API to access the raw data you have at the basis and view it in different ways. And one thing we noticed when we were analyzing these that were human genomes, when we analyzed them with some tools to map simply the data on the genome, there are several things that sort of pop out, I would say. One thing is that if we classify the pairs of, if we take each pair read and map it to the genome, one thing we can see is that some of the pairs, they just map perfectly. There is no difference to the reference genome. That's the left column here, which represents, in this case, 54.4% of all the pairs. So more than half of the pairs you have, they actually map perfectly on the human genome. And basically, it's one way to compress this, and it's been said before, you just keep the position on the reference genome and you're done. You don't need to say anything more about that pair. Then we classified those where sometimes there is an N in the sequence, and basically that's not a big deal, except that we noticed that in some run of the Illumina sequencer, sometimes for some reason all the reads in one run will have an N in one position. And it's not maybe perfect, but still you don't see any other difference to the reference genome. So we thought it was interesting to keep this category. And then the category of reads where you just, they align, but you have a few mismatches between the reference and the read. That was about a quarter of the total. And then these are reads where you need more, you need to do more to align them to the genome, like open a gap or make an insertion, and that's, in that case, that was only 4%. There, sorry. And about 15% of the reads, we just couldn't map them. Of course, if you allow more indels or more gaps, you're able to map more, but anyway, there's always some you can't map. And this category is still quite interesting in many cases, that you can, for example, be looking for, if you have an infection, that's where you might find some kind of bacteria or some kind of virus or some other thing that might have crept into the sample and could be interesting to study more, and so even though you can't map them directly, they might still have something interesting. So in our mind, as I said, some of these things are interesting because they allow you to get better compression down afterwards, but also the categorizing them can be interesting from the analysis point of view, because, for example, the reads that will show you single variants, they will come from the things that have mismatches, actually. So we thought this sort of classification might enable for and facilitate the downstream analysis. At the time we did this, the world was basically Illumina, dominated with the Illumina technology, which has used, in most cases, they use pair-dense sequencing. And we think that the fact that we have this pairing information is quite important because, well, for one thing, it represents what's actually gotten from the DNA at the first base, because to sequence, you cut the DNA in pieces, and all of these pieces, they go together. You just read them from both ends, and it's interesting to know usually how the pair was formed. It can give you some information about the length. Also, it's sometimes the pair are formed like in the chromosomal conformation capture. You actually create those pairs by thymine, and then it's interesting to know where each originated from. So it's interesting, usually downstream, to be able to keep this information. These go by pair. And when we started putting all this together, we came with this kind of block structure where, for example, for perfect reads, if we actually want to capture all the information that was in the FASTQ, and we want to encode it for the perfect matching read for each pair, we just need what was the position of the mapping. We need to keep the read headers and the read quality. But as was mentioned also before, if you are not interested to keep everything, you could have a lossy kind of compression where you could get rid of those 2 blocks if you really want to pare down to what's actually useful in the reads, because mainly the read headers are not that useful. Maybe at some, for most analysis, we don't use them. And the quality values, there are usually too many. You basically, when we did the analysis, we were just interested to set some threshold somewhere where we do actually believe what the read tells us, and when the quality is too low, we don't really believe it. So a kind of a binary thing would be enough, whereas they usually have 40 different values, which probably are not so interesting, or at least in most of the applications I've seen, it's not that interesting. And so similarly, when you need to encode reads that have also mismatches and indels, you can just add a few more blocks to capture that additional information in the form of mismatch and in the form of what's called CIGAR, which basically tell you how to place the insertions and deletions. And we didn't want ourselves to go, and we didn't feel like it was our job to start implementing a standard, but luckily across the tree at EPFL there was a group involved in MPEG, which had long experience with the compressing data and making these standards, and Claudio will present it later. And that's when we sort of became involved in this group to try to go on the path of standardizing thing. How am I doing time? Okay, so we, just to give some more background, we participated in the development of a prenatal test where you try to determine if a fetus has a trisomy or not, instead of puncturing the belly of the mother to capture some amniotic fluid, which is a kind of a dangerous process. You just do a normal blood draw, and it so happens that in the blood you have, when a cell of an organism dies and explodes, some of its DNA, and the DNA pieces get rejected in the bloodstream, like in the blood plasma. And you can capture those small DNA pieces, and it turns out about 10% of those will be coming from the fetus through they travel through the placenta of the mother. And by analyzing those small DNA pieces, you can actually determine whether the fetus has a risk of having a trisomy or not. And some studies of these, they were published recently, where we followed up on more than 6,000 cases. And this got also us thinking to do these in vitro diagnostic tests, you have to go through some normalization process, like in Europe and Switzerland. You need to have a CE marking for the software, which is considered in vitro diagnostic device. And this needs to, you need to certify that the software is operating to some criteria, etc. So we think that this normation process will also help in this respect, to be able to show that the data you process goes in a certain, follows a well-defined process. And also recently in Europe, the Global General Data Protection Regulation came into sense, and of course dealing with genomic data is some kind of personalized data, and you need to decide how you give access. And for example, if you want a doctor to have access only to a part of your genome because he doesn't need to see everything. Maybe in some cases of diseases, only a small part is necessary. So having ways to restrict those access, we think would be also quite useful. So if all this is sort of ingrained into the standard, that will ease the later processing and showing that you actually follow the rule. So the last thing I wanted to explain was a participation and where we needed to reanalyze. In this case, it was a plant genome. There is a University of Lausanne, when it was kind of the thing to do to sequence new organisms, they had an old oak tree that was planted apparently when Napoleone came to visit Switzerland, and they thought it would be interesting, since it's a very old tree, to see how its genome had diverged from the time the tree was planted to 200 years later. And if you took samples at two different positions on the tree, that's about 400 years of evolution, and the idea was to try to see how many variants you would find. And I guess I should so to conclude this, we didn't find as many as we thought we would. It was there was only a few variants, and we had a hard time convincing the referees that it wasn't because our tools were not detecting them. It was because actually there is few variants, because there are some mechanics in ways in the plants that preserve the genomes of these things. And so I'll jump to the acknowledgements. So these are the people from my group who participated in the various analyses. That's the team from EPFL who helped us prepare this MPEG, and these people were provided the data for this genome thing I just showed. And with that, thank you for your attention.
So it is time for a couple of questions while we switch between speakers.
So what was the error rate that you detected between two cells on different branches? Just one.
So we only could find 17 actual differences that we could show when we actually read it.
Is it genome?
It's about 1 gigabases. It's not so big. Yeah, and it nicely pans along. So they had I think 26 points, and it nicely goes with the variant, so you can see that they actually follow the branches. It's quite a nice thing. It was done as a sort of an experiment to also get the local people interested and the students, and to have they do a local lab thing where students can go and sequence some of that, see how sequencing is done and all this. So it's a kind of a mix between society and science, and it's quite nice because it's a visual kind of thing you can see.